Good afternoon, everyone. This is my presentation for bioinformatics component in personalized medicine, where I talk about different domains in bioinformatics: proteomics, structural biology, metabolomics, cell signaling, genomics, transcriptomics, and how each of the components in an integrated fashion or a multi-omics fashion is the key approach for solving the problems of medicine domain. So recently, not very long ago, a few months ago, we had news that 5 more deaths were found by SARS-like viruses in May 2013, and a few days later, more viruses like SARS were identified in this month itself, July 2013. So how do we combat such difficult problems? How do we immediately solve such problems? Can we wait for 1 month of experiments to be done, or should we do it quickly by more logic, computational methods, and software? So this is kind of a review of the first session I'm talking about proteomics, structural biology. So in 2013, we had the SARS virus outbreak. It was supposed to be originated either in Toronto or in Hong Kong. What is the case be? So looking at the morphology, the virus belongs to the coronavirus, the virus category. And so the coronaviruses are not that virulent or harmful as this virus was coming out doing. So there got to be something different in it which we were not able to say just by looking at it under the microscope. So the key way is to look at the molecules or the biomolecules in it, and the genome was sequenced quickly. Luckily, the genome was not that big, as is the case in viruses. And all the different genes were identified by mapping it to other coronaviruses. And certain regions, known as X or an unknown, there were many X's, were left unidentified for their functionality. So obviously, what are the virulent factors that we have in this virus can be attributed to this unknown, these unknown genes or ORF. ORF stands for open reading frames, different frames in which the DNA gets transcribed and translated. So this ORF was taken and then blasted or put in a blast comparison to this NCBI database. And the best match we got was a calcium binding protein. What do we do with this match? We then thought, let's look at the structure of it. And the structure was not available in the database because it takes a lot of space to generate a valid, good structure of a protein. So the structure was not available, so what do we think logically? We took this match and blasted it again and found other calcium-binding proteins of that domain and picked up those ones which have their structure in the RASMOL or PDB protein data bank database and did a multiple sequence alignment and found out the regions of homology. Star is a perfect alignment, or the cell colon would mean either physical or chemical similarities in the amino acid residues. So once this homology is collected, we can take the consensus with these motifs and build a structure of it computationally by prediction. Go to the lower energy levels and all those things. And once that structure is predicted with certain confidence, maybe 70%, whatever best we can do, protein-folding problem is an unsolved problem yet. Then we look at different cations which can bind to that whole homology region, which are the calcium-binding protein. And we found that this ORF also binds to calcium. And it shows it can also bind other cations like magnesium or whatever is in the periodic table for the calcium in the row, in the column. But the best energy level was coming for calcium, and we proposed that this protein is a calcium-binding protein, and essentially a virus that it introduces its genome into the host, such as human beings. Wherever this calcium binding is important for the metabolic functionality, there it will take away all its functionality and can eventually lead to death. There were also some more analysis such as the transmembrane region as to where does it exactly bind and how the trafficking of the metabolic can take place. So this is the power of, and eventually in 2003, this was published 2004, and eventually the SARS outbreak was stopped. But there were many more researchers doing this. So then I move to metabolomics and cell signaling. So what is metabolomics is that if we have a urine sample or a blood sample or any human body fluid sample, if we can take out its peptides or proteins that are being secreted, then we can look for the signatures in them and match it to a database of known people with diseases. So say someone is suffering from certain kind of cancer and he expresses certain kind of additional peptides during that stage, then for a person who wants to get himself diagnosed, we can look at those samples and do a comparison. And if the signature matches, we say, okay, there's a certain possibility that you're at this stage of your illness. So what is done is that peptide chain is there, we break it for lysis into the region, we charge it, so we get an M by Z value, and then we have a plot of dilution because we're not sure whether that M by Z value we're getting it right. So we dilute it, and so we get a certain curve. And now this curve is so disturbing, like it sometimes goes up, sometimes goes down, and it's not so regular. So again, there's a need of bioinformatics to treat each of these curves individually, reduce the noises, come up with the least regression of plot, and then generate a weighted and measured value, and then come to the comparison. So this is all mathematical, computer science, logic that is involved. So I'm here to talk about proteome break. So the blue lines are the actual data plots. And this is the red line is the one which you will get when you do not reduce the noises, do not do mathematical analysis. If I cut down the noises may not necessarily be the front, it can even be the back. We generate this regression line, the green line. Such a big difference in the slope of these lines would generate such a magnitude of difference in the resulting M by Z value that we take into consideration. We can also opt for a non-linear regression line, such as the supplement. This was written in MATLAB, and they also made a standalone Java version of the software. So this can be hooked up essentially to the database, and we can find out the stage of an individual. And for example, if we in a cell, if you know a certain pathway, such as the Wnt signaling, which is important in cancer or even in actually development, and if this development process goes wrong, then a certain section is overproduced. We have unwanted proteins, then that leads to cancers. So all these reactions, these are all chemical reactions. They are happening inside the cell, we call it biochemical reactions. The chemical kinetics law will be observed here. There are certain, tuned in chemical kinetic laws like Kn, Kcat, forward, fast, forward, backward. All these parameters can be fitted into a reaction mathematically, and then we can plot the way all these components can plot it, how their concentration varies with time. So this is without any external signal to the cell. And once the signal is changed, how the variation of each of these components can change. And that's what we have is a virtual cell or an artificial cell that we have generated. We can control the cell now. Or if the person is suffering from certain things, we can do things that are experimentally legitimate computationally and have a closer understanding of what's actually happening inside the cell or the diseased person, as the case be. So another case would be like if we have a set of reactions, chemical reactions, biochemical reactions like this, where the downstream product goes and binds to a certain pocket in the enzyme such that it affects the catalytic side, the reaction side of the enzyme, and thus the reaction can either stop, reduce, or it may be enhanced. What is the case be here? So if it stops, we'll have less production of that, the ultimate metabolite that we are interested in, and that might be not what we want. For example, if we want oil, lipid, then we don't want this regulation. We just want to cut off these regulations, and this reaction will just boom in. Go forward more and more. And on the other hand, when this is going very, very fast forward, we want to stop it or reduce it. Such as in the case of cancer, when Z is like overproduced, we don't want that to happen. We want to stop this. We want to regulate this because feed forward, it is enhancing. We want to stop it. So what I proposed is that we make an artificial chamber like this, and it's selectively permeable. C goes out selectively. None of these components can go back in, but only C can go out. So this will happen. But can this actually happen in a biosimilar environment? The cell multiplies and divides, so will this compartment also multiply and divide in the next daughter cell? Not really. So what we do is, physiologically, instead of going, so essentially if we wait for 1 million years, theoretically, all the allosteric domains will go away. As per Darwin's law of survival of the fittest, selection pressure and stuff like that. So after 1 million years, the allosteric domain will go away, only the magnetic domain will remain, and if we remove this chamber at that moment, we just have the desired reaction without any allosteric control. We have a deregulation here. So we can't wait for 1 million years forward. What we can do is go 1 million years backward phylogenetically. So we go for the same reactions, we find for the same reactions in a different species where such thing has happened naturally. What I'm supposing is done. It's not possible to wait 1 million years. We go for 1 million years backward, and thus we can have a bit of control in cancer, as I said before, overproduction of vitamin B5, phytosinase, which is an end product in itself, or it can be hooked up to the lipid pathway or production pathway. So the phytosinase, this lead compound binds here, and it regulates it. So I can make a compartment here, typically, and we have an unregulated control. So coming to the genomic section, which is the final section, I already talked about it in detail earlier. I'll talk about it again very briefly because maybe there's some more new audiences here. So we know that the variations can be broadly classified in SNP and structural variations, which can be further important indels and complement variations. And there are different methods that are already existing: assembly approach, split read, read depth, read pair, because we know the read length can iterate from the fragment, we know the insert size roughly, but if when we map this whole bit to a reference, and if this information does not exactly match what we would have expected, then there is a insertion deletion, and if it doesn't match the number of coverage we did in experiments, there can be a duplication or a complement variation. So these are very logical, and the thing is how do you put it in the form of a program for data which is of the order of terabytes. We are not small data of megabytes or gigabytes, we have terabytes of data, or maybe at least 200 gigabytes. So that's the challenge. Computational complexity, time is important, data handling is important, and the logic is the most important, the algorithm. And this will be the end result that we are interested in certain loci region, which might have a gene which is responsible for certain functions, such as for a person which is linked up for a person to get taller, or a person to have sickle cell anemia problem. We can look at all those factors that are there. And I made a software called Genome Break, which can find out variation from as small as just one basis to as big as 1 million bases. That's a huge number, 1 million bases. And so we can also find a sex determination of a person by taking out the DNA from the fetal state of an organism or human being, and determine the sex of the individual by just comparing the matches with the father and the mother. A stands for father, B for mother. C and D are two children which happen to be the daughter in this case. And this was more deterministic than I used the structural variations instead of SNPs, as there's a relative difference both higher. And it also proposed that SVs are higher, better characteristics, signature of an individual phenotype that we are interested in. And it also says that SVs have more restriction in terms of the selection pressure and changes that are there. And that's obviously because codons that code for amino acids have like, there are multiple codons that code for the same amino acids. So SNPs can have a lot, yet code for the same amino acid, because the selection pressure doesn't change it, but when we change the indels, there is a frame shift, there is a codon shift, there is a the amino acids can change, a lot of things can happen. So there's more selection pressure when structural variation happens. So the conclusion is we saw that all these factors in a combined way, the bioinformatics is very crucial. When we talk about proteomics, genomics, cell signaling, metabolomics, there's an integrated approach to solve the data problem. In our case, it's a medical problem. These are a couple of my papers. Actually. That's my email ID, and that's my page to get back to me. Questions? Can go to slide number 10. Expensive. Sure. Yeah. So, yes, right. What kind of signal process do you expect to how? One more slide. I see. Earlier. So this is all interconnected reaction. So there is a cell, and here is the beta catenin that is formed. And all the reactions are numbered from 1, 2, 3, 4, and all these things. So Wnt is the first signal that comes in. That affects this reaction. That affects in terms of all these reactions, and ultimately beta catenin, which is important for the research and development process. So whether externally we get a Wnt signal is there at the cell surface, if it's present or it's not present, and I think it's the gradient of presence, that doesn't matter. The cell does not respond to presence or not presence, it responds to the change of presence. The delta thing is there, that is important, which gives a signal of value. So in my case I just took 0 and 1, but in reality actually it can be a number between 0 and 1 that is involved. Is there any way can we control or module or change? Yes, that's what I mean. It's a virtual cell. So you know that how the metabolic profile is changing. The yellow line was like this, it changed like this. The red line was like this, and almost 0 and 3 of them. They all grew to a certain extent with time. But this is a simulation, right? This is simulation, and it was also experimentally confirmed by our Harvard collaborators. This work was done in Los Angeles National Lab, and we have Harvard collaborators. And what we simulated, the software is called BioNextGen. What we simulate for Wnt signaling for this pathway Wnt signaling was confirmed completely by our experimental collaborators. So that means we have more faith in our software, and eventually, even if we have no collaborators later on for experiment service, we can do a virtual analysis. Okay, very kind.